Buonasera.
Good evening.
Buonasera a tutti, benvenuti, bentornati.
Good evening to you all. Welcome back to GiovedìScienza. Science on a Thursday. As always, we have two gong players and we will try out mic and gong. So to your left,
Arrivo
Lara.
dall'Università di Chimica qua di Torino.
And I'm from chemistry here in Turin, and I'm quite interested in the medical field, and so I wanted to take part and hear something new.
Stefano, neolaureato in biotecnologia medica.
My name is Stefano. I've just graduated in biotechnologies, one of the first conferences I'm attending as a new graduate.
Uno dei due funziona anche meglio.
Yes, it works.
Può dare di più. Benissimo. Allora,
fine, it works even better. So I would like to remind you, and in particular our guests, that the two gong players can interrupt the conference at any point to ask a question, one question each, or to ask the speaker to illustrate a point more in depth. I too will be sitting. We have a science journalist. Yes, I'm just a trainee, Piero Bianucci.
E invece alla mia sinistra c'è
to my left, Michele De Luca. Round of applause for Michele De Luca.
Michele De Luca è un medico.
Michele De Luca is a doctor, a medical doctor, first of all. He is a chair at the University of Modena and Reggio Emilia, who at a certain point decided to switch to research. Last week with Pierpaolo Pandolfi, we finished talking about medicine, so we can continue talking about medicine, and here we have various levels. This evening too, we will be talking about innovative treatment. We will be doing it with an Italian doctor, medical doctor, who is famous in Italy and abroad, and so it should be, and we will be talking about research but also treatment. So I would now like to ask Michele De Luca if it's true that you started working as a hospital doctor, as a registrar. Yes, I'm a specialist in endocrinology, so after my degree I started working in Rome, at the University of Rome, on thyroid. Yes, I saw several people every day, several patients. And what happened at a certain point? I was bored to the gills. I was not really stimulated by what I was doing, so I decided to leave everything, thinking that I hadn't quite made a mistake, but that wasn't what I wanted to do. And I left for the US, where I started working in the field of research. There I met, in Boston, Howard Green, who was my master at the time. He was establishing the stem cell laboratories, which at the time weren't even called stem cells, and I fell in love with the system. Then I worked as a researcher, but with a clinical eye. That is to say, to translate fundamental research into clinical results, especially for rare diseases or for diseases that haven't got an alternative. So the fact that you're a doctor was important. Oh, yes, absolutely. Piero, you've known Michele De Luca for a long time. Yes, I've known him for quite a while, and I would like to say that he is here, as was Pandolfi last week, thanks to Fiorella Altruda, who is our chairperson of the Scientific Committee for GiovedìScienza Science on a Thursday. But the first time that we met him was a few geological eras ago, when stem cells, that we were starting to call stem cells, were still a glimmer on the horizon, but he was already exploring them. And Piercarlo Marchisio who was a professor at the University of Turin, and then at the San Raffaele in Milan, and then again working with De Luca in Modena. He was the one who spoke about this promise in research. And the second and last thing I'd like to say is that then we had a virtual meeting on a day about a year ago, and with the Ferrero Foundation on good aging, and as we now call it, De Luca has written hundreds of papers on stem cells, but De Luca he couldn't come that day because there was an article on Science that had been released that day, and he couldn't come because at the university in Modena he had all television and newspapers. He'd done something which he would be talking about. He had treated one of those butterfly children, that is to say, he'd been a doctor. These are children that basically cannot be exposed to the air. Well, he will be telling you about it. And we saw this child playing football. I don't know if we'll see him playing football again. And there was this Skype link without the physical presence of De Luca. But the interesting thing was that there had been a clinical case that had been successfully treated, because now this child is like any other child. But in the meantime, he had made a discovery, fundamental research. He discovered something new in stem cells. So it's an incredible case where clinical studies or clinical research and fundamental research went hand in hand. And this explains why a clinical case ended up on Science, because usually Science doesn't publish anything like that. But in this case, it was both. So I'm very pleased this evening to have the opportunity to hear Michele De Luca, and I would like to thank Fiorella Artruda and Piercarlo Marchisio for having brought him here. So we will be talking about stem cells, gene therapy and regenerative therapy. Give him the floor. Thank you very much. Thank you very much. Well, thank you, Fiorella. We are involved in regenerative medicine, which is a new part of medicine. 15, 20 years, the aim of which is the fact that you recreate a tissue which is ill, different from surgery, different from classical medicine, and to be able to recreate a tissue, we need stem cells. Why? Because most of our tissues renew. We change our blood every 3 to 4 months. Our skin, epithelium, we change it roughly every month, and we change our gut lining every week. So we need stem cells. Very often there's a mistake. That is to say, people think that there is one stem cell that does everything. There are lots. But embryo stem cells, embryonic stem cells, and somatic or adult cells, they have different roles. For instance, the embryonic cell is also called a pluripotent cell because it's the only one in nature which is able to create all tissues and organs. However, it exists for a very limited period of time. Let us talk a bit about embryonic stem cells. Do you know what this is? It's an egg cell which is attacked by sperms, by spermatozoa. When you have fun, you have a bit too much fun, this is what happens. That is to say that one of these little animals, usually the one which is a bit more tricky, manages to penetrate the egg, and this gives rise to a zygote, where you have both genetics, and you then form a morula, and the cells are called blastomeres. this is long before the implantation in utero and the womb. And before this happens, they start differentiating. That is to say, they form a blastocyst. As I was saying, we're very far from the fact that they are going to be attached to the inside of the womb. For example, these will give here a group of cells that will give rise to the placenta, and these other cells that are within the blastocyst that are the so-called embryonic stem cells. Now, these are the ones that in the course of development will give rise to all the tissues that make up our body. There's also another way to form blastocysts, which is what you call IVF. There are many couples who, for various reasons, although their gametes are fine, are unable to have children. That is to say, to trigger the process. Science, over the past decades, has given them a hand. So now it is possible to take the oocytes, human oocytes, then fertilize them artificially in vitro, introducing the sperm in the egg and forming a certain number of blastocysts. Usually, you create more than one. And then the gynecologist, by looking at them, can see the ones which are best suited, because not all are suited, and puts them in utero, and this gives rise to a pregnancy. You may not know, but this is something that nature does. There are, for example, there are miscarriages simply because the blastocysts were not well suited. In the case of IVF, it was discovered by Robert Jeffrey Edwards. He won the Nobel Prize for it. Clearly, there was an opposition. The Roman Catholic world and the Vatican in particular said that the Nobel Prize shouldn't have been awarded to him because all ethical issues were ignored. Here I would like to say that I'm also chairperson of the Luca Association for the Freedom of Research, and say that we disagree, because Edwards' technique, which is IVF, has meant that in the world tens of thousands of children were able to come to the world. Over 1,000 in Italy every year, children that wouldn't have been born because that couple, say, was unable to have children. So it's very ethical. What happens with the other 432 blastocysts that are not used? They're frozen, and they stay in the freezer, and they can't be used anymore. So they are left to, quote, die, unquote, in the freezer. They are inappropriately called embryos because the blastocyst is the first stage of development of an embryo, but an embryo develops after it is in utero. In slightly more enlightened countries where there is more freedom, these extra blastocysts are used for research. They can't be used here because the last part of one of the worst laws approved by the Italian Parliament, Law 40, which makes it impossible for us to use blastocysts for research. But in Milan, we have Professor Cattaneo, who works on embryonic cells, human ones. Is she outside the law? No, because we can't work on the stem cells, the Italian embryonic cells, but we can import foreign ones, which is a beautiful but frightening example of hypocrisy, because two are the options. Either it's wrong and you can't use any and you can't import any, and if not, it isn't. So then we can't understand why we can't use Italian ones. Well, let us see why. Clearly, in the collective imaginary, if I say the word embryo, all of you think of this. Is it true or not? Yes, of course, this is an embryo. It is about 5 or 6 weeks old. This will not form outside the uterus. No uterus, no party. And no delivery, because in Italian, delivery is parto, which sounds a lot like party. Let us see what happens before it's implanted in utero, which is the blastocyst. The embryo is an individual in the etymological sense of the word. Because of those of you who study classics will understand me, because it's an individual because I cannot divide it. But both morula and blastocyst are not individuals because I can divide them. Each one of these blastomers can be taken out of the morula and it may generate identical individuals. They've done this with sheep, cloning lambs, and others. So we can take these human blastomers and have 32, for example, Di Maio's, who is currently our Deputy Prime Minister, 64 Salvini's, the other Deputy Prime Ministers, and 128 Renzi's. Already I had doubts with lambs, but embryonic stem cells means that we can produce chimeras. If we, for example, take some blastomers from a white mouse and put them in the blastocyst of a black mouse, we will give rise to a mouse that will not be gray, as you might think. It is an unbearable, unlookable, striped mouse, black and white. Now, I vie for the Inter football team, and therefore we hate the Juventus black and whites. Now, why do I get angry? Because these cells can give rise to all the tissues, and research has now proved that by taking the stem cells, the embryonic stem cells, and putting them in culture, we can generate any. I'll give you some examples from enlightened countries where this is leading to a treatment. This is a retina. This is the optical nerve. This is the macula, where we have photoreceptors. We can see these two children with footballs clearly because this system works. The average age of human beings has gone from roughly 40 to 85, thanks to science. But this means that there is an increase of age-related disease, and this is an age-related macular degeneration, AMD. Now, this might be different types, dry and wet, but which progressively leads to a blurring of the visual field, which gets bigger and bigger, and these people progressively become blind. I have some friends, a couple were artists who had to discontinue their activity because they suffer from this disease, which is irreversible, progressive, and incurable. What did my friend Pitcoffey in London do with the London Eye Project? He took stem cells, embryonic stem cells. He generated retinal cells. Look at them. They're even more beautiful, the ones which are derived from RPE, than an adult one. He produced a patch which was made up of the stem cells, and he implanted them in 2 patients in a clinical phase 1 and phase 2 experimentation or trial. This is one of the 2 patients. This patient here couldn't see the book he was holding, and now he can read it. Can you tell me, in a non-religious country, is it more ethical not to use these stem cells that will never give rise to a human being, or if it's more ethical to use it to give this person back their sight? I'll tell you more. I'll add something. There is a disease which is called Parkinson's disease. And it is one of the ones that can be attacked more easily, because this group of cells that are in the black matter of the brain is a group of increasingly specialized cells that give the signals to the striate where dopamine is produced. In the absence of these, we have Parkinson's disease, which is a very severe disease. What has a Swedish group done? What did they do years ago? They took fetal dopaminergic neuroblasts and injected them in the striatum. I'll just show you an example. This was the patient before the implant. And this is years after they've had a transplant, and there are studies that have gone on the study of these fetal dopaminergic injections. What's the problem? The problem is to do this, we need routine abortions, consent. We have ethical guidelines. We have donor age restrictions. We also have 3 or 4 fetuses that have been aborted per patient. You can't imagine that you'll be treating Parkinson for the whole population, not only, but there will never be the possibility to standardize these procedures to have a reproducible cell treatment. What is Lawrence Studer doing in the US and Malin Palmer in Sweden? They've taken the embryonic stem cells, got them to differentiate and produce these spectacular dopaminergic embryos. In fact, in 2019, they will start a phase 1 clinical trial in the US and in 2020 in Europe. And if these are the results with the fetal dopaminergic, you can imagine what a standardized preparation of dopaminergic characterized production by the stem cells, you can imagine what they can do. I repeat, what is more ethical? What does ethical mean? Let us not confuse ethics with religion, please. Having said this, we then have somatic stem cells, which are the adult ones that are inside an adult body. Blood, skin, cornea, intestine, or gut. They're all stem cells that are specialized. Instead of being pluripotent, like the embryonic, they are oligo or multipotent because they only produce the cells of the tissue where they are. Skin produces skin, blood produces blood, and so on. So what is a somatic stem cell? It is one which is responsible for the generation and regeneration of that tissue. If we didn't have a blood stem cell, we would die because we wouldn't have any blood in 3 months. We would lose our gut, we would lose our intestine, we would lose our skin. There are induced pluripotent stem cells known as iPSs. This is slightly more technical. That is to say, by genetically manipulating things, it is possible for a somatic cell to become an embryo-like or embryonic-like, which means that they don't have any problems of immune suppression. Of this wonderful landscape, what do we have in the clinical level? Well, we have the first clinical application was blood. This gentleman, who was called Edward Donald Thomas, who understood that the Japanese that had been bombed in Hiroshima and Nagasaki died because of the radiation, and understood that by destroying the blood of leukemic patients and then reinserting it with a bone marrow transplant that contained hematopoietic cells, he could save the lives of these children. Now, this is regularly used to transplant, and it saves, if I'm not mistaken, 80% of all leukemia patients that would otherwise die. The first clinical application of a cultured stem cell, because normally you don't. You just aspire it for the bone marrow. It's an aspiration and then reinjection. But if we talk about ex vivo, that is to say recreated in a lab, my master and mentor, Howard Green, in 1984, that was the official, from my point of view, official beginning of regenerative medicine as we know it today. At the time I was working in his lab. Don't start working out my age, because I was just a baby at the time. I was a baby. But I was very precocious, yes. He saved 2 children who had 90% burns, transplanting them with cultured cells, their cells, that had been transplanted on these children. And this is what we are involved in. And I'll show you some examples, which are success stories of stem cells. This is a piece of skin. If we take the skin and we isolate the cells, can you see these red blobs or gray ones? These are colonies of cells. Each one of these colonies is the progeny of a single cell. At a certain point, these cells merge, form a continuous layer, but as you can see, there's an enzyme which can make it possible for this epithelium to be detached from the culture plate. This is a piece of skin, and it was the first tissue to have been recreated in a lab. Our PhD students or our postdocs are used to seeing pieces of cartilage, bones, and neurons that are generated, the organoids. But at the time, the mid-80s, to see a human tissue that emerged from a culture plate was, I assure you, it was emotional. These cells can also be cultured on fibrin, and they can be used for cell and gene therapy. I'm going to give you 2 examples of each. I'm not going to talk about burns, because this is part of history, but thousands of patients were treated, as Green did in 1984, with third-degree ones. And in Italy, too, when I re-entered in 85, 86, we did a few hundreds of these, and many here in Turin, in cooperation with the CTO hospital. At the time, it was Maurizio Stella, who was very young then, and we treated several patients in the beginning of the 90s, in the early 90s. Let me give you 2 examples. One is the cornea, and in particular Graziella Pellegrini. I've been working with her for many a year in cooperation with Paolo Rama, who is a San Raffaele eye surgeon, very good, and I'll show you what we do. This is the cornea, this is the conjunctiva, 2 different types of epithelia. The stem cells, the corneal stem cells, are in the limbus. It is a border area between the 2. We have characterized it. I'm not going to talk about the details, also because it's not you're not a technical audience, but these treatments have to require a lot of research, You have to characterize these cells. You have to know what we have We also have to check what their ability to proliferate, differentiate is, and then you can work with the patient. Not only, but the patient is able to answer, is able to help us understand whether what we have understood of the stem cells of that tissue is true, because patients' results can't lie. When we have a central burn, a chemical one, for example, a chemical burn, and we have this area called the limbus that is still preserved, we do what is called keratoplasty, that is to say, a transplant of the cornea. And it works very well. It works fine because we also have on this eye a population of stem cell from the limbus so that it's able to recover and to produce a new epithelium on the transplant. But we have eyes also like this. Eyes where the burn was so strong that it destroyed the cornea, the central cornea, but also the outer area. It's white. Why is it white? Well, because the eye defends itself and makes the conjunctival cells migrate, and it becomes a pannus. So that is to say, it's all a conjunctiva. And it's a conjunctivization and loses the ability to see. In this case, the cornea transplant can't work because we have a complete destruction of the stem cells. After we have characterized all those cells, as I mentioned before, we thought that we could take a small biopsy, just 1 or 2 millimeters, a biopsy. We took them, we cultivated them, we grew them, and we worked on this for one eye or both eyes, as long as there was a small limbal area. I won't show you the surgical procedure because you'll faint, but I'll show you the eyes before. You can see how this eye is opaque. It's opaque here. This is a failure. And look at them after the work that the stem cells did, how the eye becomes normal again. It's no longer vascularized, so the cornea is transparent. These eyes have a full recovery of visual acuity. See other eyes before and after that basically say the same thing. Right. This is a bilateral case with corneal regeneration. This was a youngster who had a burn due to quicklime and had various transplants. He couldn't drive, he couldn't work, but from a biopsy we had 2 transplants, full recovery of visual acuity. This therapy, now I forgot one slide. This, as I said, over the years has become better and become much better, especially in the last 15 years. Graziella Pellegrini, we've been working together for many years, has been registered. It's now called Holoclar, and EMA, the European Medicines Agency, recognizes it as a treatment, and it has been approved in the Western world. It comes from Italy, the same country that can't work on stem cells and embryonic stem cells. Once we have the stem cells and we know them, we then know how to apply them in a clinical context to have tissue regeneration. We can then imagine or decide to carry out gene therapy, that is to say, to modify them, because a burn, both of the eye or of the skin, is, well, it's damage by an accident, due to an accident, fire or chemical, but we have genetic diseases where these cells are altered. And in the field of blood, for example, which was the first stem cell to be applied is another Italian result. From the San Raffaele in Milan, Carlo Borgognon, Alessandro Iudi, Luigi Dandini. That is to say, working on modification of hematopoietic stem cells to treat, for example, immune deficiency due to adenosine deaminase deficiency, or even other rare genetic diseases, such, for example, metachromatic leukodystrophy. Now, clearly, the second stem cell that was used in gene therapy was the skin one, or epidermal one. We have a big problem, which is the so-called butterfly children. I'm sorry because of the images, but this is what it is. It's inherited epidermolysis bullosa, known as the butterfly disease. Minimal trauma, touching simply. So you have these lesions that are formed, blistering. You can imagine that this child cannot have a normal life, can't be touched, can't be embraced, can't play, can't spend time with other children. You form a sort of bubble around them because every trauma in the severe forms cause bullous lesions. Short life expectancy. 40% of the junctional children does not reach 18. Most die in the first 2 years. Those who survive their childhood have a very short life expectancy, while because they develop squamous cell carcinomas, very aggressive ones. Clearly, that since it's a genetic disease, the only real treatment that you can have is to treat the gene, that is to say, to treat the genetic disease. Now, first you must understand which genes are involved. Our epidermis, which is attached to the derma, you can see them here, by a series of proteins, one attached to the other. Keratins, the basal one, this interacts with the integrin, which about 82 years ago, it was 90 years ago, yes, Marchisio and I proved what the receptor was. They interact with the laminin 5 and with collagen 7. So this anchoring chain means that it keeps the epidermal attached to the derm part. If they're not, you have these bullous lesions forming. In 2006, we carried out a clinical experiment on our patient called Claudio, who was the first ever treated with a gene therapy ex vivo, and we transplanted 2 legs because he had a junctional form linked to this problem. It tells us that the result was that he was better. He was treated, cured. How do you do gene therapy? You use nature. Viruses infect cells. We know that. So, if we take a virus, we modify its genome, and instead of his genome, we put the gene that we're interested in, leaving in the virus the machinery or the system that it uses to infect the cells. The virus, which is stupid, infects the cell and transfers into the genome of my cell the gene that I want. So by modify this, I can put a correct version of the missing gene and therefore create the correct protein, which is what we did with Claudio. And his skin got better in the legs. So why did we not do other patients after 2006? Why didn't we do other areas of Claudio? Well, I'll tell you very quickly. We didn't do this because in the meantime, in 2007, the law changed. The European community had new rules for advanced treatment. Fiorella Truda knows that very well because she created a cell factory in Turin. So all these treatments must be done in GMP. That is to say, you have to create some cell factories. To be consistent with the law, you must interact with the drug companies because the type of skills are not in universities but in drug companies. We did it in Modena. We created the GMP. We interacted with the Chiesi group that were interested in carrying this. We founded a biotechnological company that is called Holostem, and all this enabled us to continue with our gene therapy. In the meantime, we wasted 9 years, lost completely, to be able to develop these treatments. And as I told you, in the case of Claudio's legs were quite promising. So in 2015, we managed to resume gene therapy. In 2015, something happened which in some way gave us back part of the time that we'd lost. And that's a very beautiful story because it helps you understand how beautiful science can be, how beautiful fundamental science can applied to clinical studies can be, and regenerative disease, which since it's a new field, it can put all the various expertise together. You need the biologists, molecular biologists, genetic experts. You need everybody working together. And have I still got some time? Yep. I was forgetting something very important. In 2006, Oxford, the Oxford Dictionary, it chose post-truth as the word of the year. Post-truth indicates that condition whereby in a discussion referred to a fact or news, truth is considered a matter of less importance. Does this tell you something about what is happening just now? That is to say, in the post-truth, news is perceived as true by the public, by the audience. So, which means social networks, means those who are in government, on the basis of feelings without any actual truth of facts. So, the feelings, the emotions, the personal ideas are more important than facts, even in political decision makers. Beware, because this is an attack on a scientific method. Big democracies have always had a very strong drive for research. They've always emerged from crises through research. And if our politicians were to use a scientific method to consider and assess facts before making decisions, they wouldn't fall into that sort of functional illiteracy which they have, because this is what they is, and which is attacking us. So an attack on the scientific method has an impact on society. Think of vaccines. And think of stamina. Think also of the block of research on embryonic stem cells. Stamina is a perfect example of information on Facebook which is not true, which television programs, such as the hyenas, can say without any scientific benefit. And Davide Manoni from Moncalieri near Turin has damaged this country because he is the example of pseudoscience and of what science should not be, because it should be based on facts. I'm telling you this because unfortunately, in the field of stem cells and regenerative medicine, there is a tendency to have a few short circuits, to not have strong fundamental research, not proving in facts before you move on is something which happens.
L'altra volta in chiusura forse c'era stata una
We
domanda.
haven't had
Gonghisti
any goings. This is the second time. Well, the other time, maybe there had been a question. I'm going to ask them now. Have you a question? No? Because it was all very clear.
Ringrazio personalmente per la bellissima conferenza e anche per gli agganci
Thank you
non
for
solo
the
scientifici
very
e non solo
beautiful
tecnici.
conference, very interesting, and also for the scientific and not only scientific ideas that are paramount. My question concerns the treatment of glaucoma. I know that there are experiments in the UK, in London in particular, and some experimental results have been obtained. There are also initiatives in Italy, liquid surgery in Mantua, and other research projects that concern CRISPR-Cas9 on as far as the genetics are concerned. I would like your opinion on the state of the art. Well, with an advanced treatment, that is to say, with this type of treatment for glaucoma, as far as I know, is not available. But I do know that there are various groups, as some of the ones you mentioned, that are working in different directions. Unfortunately, the issue is true for cornea, for skin, for blood, for cell treatment, but science needs time. So it's still early to be able to say it, in particular with this factor that these advanced treatments are regulated also through a number of drug procedures. There are a number of tests and trials which are there to protect patients. I'm sure that something will come out. I know that there are several trials underway from different points of view on glaucoma, and as far as I know, none so far has been able to solve the problem.
Scusi, io conosco una società criogenica, no, di ibernazione.
Please excuse me, I know a cryogenic society of hibernation, and they too are talking about nanotechnology. That is to say, molecular manipulation of the brain, and so on. What do you think of nanotechnology and of genetic engineering? Well, in genetic engineering and nanotechnology are increasing in serious things. They're doing incredible things from this point of view, both biotechnology and genetics, gene therapy, hibernation. Please don't ask me to comment on hibernation and cryogenetics, please, because I've said enough about other things. There's another question.
Tutte le spiegazioni, la cosa che più mi ha entusiasmato,
the thing that I was most enthusiastic about is that through the metaphor that you used, I finally understood how the modified virus enters the cell and releases the healthy gene. Now, what I would like to know from you is, where's the problem? Where's the difficulty? Since now we've had this young swimmer, for example, he's had an injury of the back spine and the bone marrow. Why can't he be treated? That's something different. What regenerative medicine does is to have a very clear objective with a very clear rationale on a very specific disease with a specific stem cells. everything has to work like clockwork. Let me give you an example of the brain. Why are we trying to work on Parkinson's? Because if cell therapy with dopaminergic neurons that come from embryonic stem cells were not to work on Parkinson's for a number of reasons, imagine what would happen with the others, because Parkinson's is simple. When you break your back and you have your bone marrow injury, think of all the work of the neurons. It's not that simple. You have to contextualize things. Why, for example, blood and skin or cornea were, in quotes, easy? Well, because they're simple tissues. Blood is a liquid. You've got to just put the stem cell, and the stem cell finds its way and regenerates the tissue. Skin is a bidimensional one, but my friend Giulio Coso is going mad to try and replace the muscle in Duchenne's muscular dystrophy, because it's not that simple. For me, it's simple to take the skin off and put some new skin like it is for cornea. A myeloablation is very simple. But how can you develop a muscle? It's very difficult. So the more complex the tissue or the organ is, the more difficult it is to have this solution. You could also have the right stem cell in your hand, which again, for the time being, we haven't got for motor neurons. So in any case, we haven't got it. But we've got one for a muscle. But if you're going to transfer it into the clinical sphere, it becomes a problem. So one thing is having skin, but another thing is brains or kidneys or livers or heart. The same is true for motor neurons. I didn't say that we won't get there in the future. I don't know what will happen in the next 100 years, but these are enormous hurdles. I think that in the end, we will also have these more complex tissues and have this through other routes, not only using the ones which we are using now that are a bit barbarian, so to speak, but you need time. Another question?
Buonasera. Io volevo sapere,
Good evening.
ma sul
I would like to know,
non c'è
but
proprio
as far as
nessuna
Parkinson's is
possibilità,
concerned,
di
is there no
pensare a
possibility
una cura, un
of
trapianto,
thinking of a
almeno
treatment or a
se non su tutti i
transplant,
malati, almeno su quelli molto
if
giovani?
not on all patients, at least on the young ones? Well, that's what they're trying to do. They're trying to... Well, Parkinson have its treatments. It's not that Parkinson's hasn't got any treatment. You don't cure it, but you manage it pharmacologically, and it's progressive. But as far as the advanced treatments, the ones we're talking about with stem cells, it's exactly what they're trying to do. They're trying to... Lawrence Studer, for instance, from the USA, and Malin Parma in Lund are trying to select, according to a number of criteria that are called inclusion criteria, they're trying to select the patients that have a better chance of reimplant of these neurons that might work. The only way to do it is clinical trial, phase 1 and phase 2, which is the one that both Palmer and Student will be doing next year. Are there any other questions?
Io mi scuso se non ho
Please excuse me if I haven't got the right words because I'm only a housewife. But I wanted to know if there is a stem cell that can reconstruct the bone of the gum, because for the past 2 months that the dentist has taken teeth out and there's still pus, and they say that there is a beginning of necrosis here. Thank you very much, and I congratulate you because I too understood various things, and I'm not very well educated. Your question is very interesting because it gives me the opportunity to say something else, quoting fundamental research by many people, including Paolo Bianco. The cell that you are talking about exists, and it is the one which is wrongly called mesenchymal stem cell. In the bone marrow, apart from blood stem cells, there are these stem cells that are mesenchymal, although mesenchymal is not the right term, but let us call them mesenchymal, that represent 0.1% of stromal cells and that have the ability to create a bone in another cell. For pieces of bones, they can ossicles with, in fact, inside the bone marrow component, which recalls stem cells and can also lead to hematopoiesis. So the cell exists, is well known, but we still haven't found the way to transfer it into clinical trials. We've carried out experiments, clinical trials. One of the first, for instance, in Italy was Professor Cancedda many years ago for the recreation of the bone. There is still a number of problems that have to be solved, but the stem cell that Paolo Bianco calls the skeletrical and not mesenchymal stem cell, we have to find the way to transfer it into the clinical environment. So if I've understood, you've managed in a lab to develop these ossoids or ossicles, but you haven't yet tried to transplant them into the patient. Some attempts have been made on animals, also in human beings a while ago, and they're not very convincing, and we still haven't found the ways to replace parts of bone. But the cell exists. There's still a question.
Intanto grazie per la conferenza, è stata
thank you very much
veramente
for the
bella
conference.
Io avrei una domanda
Clear and very interesting. I have a question of the insertion of the gene during gene therapy. What method did you use to insert the gene, and also if there were any problems on the correct insertion where you wanted it to be inserted? Were there problems of interference with other genes and so on? No, we can't, apart for some molecular biology techniques that are underway, gene editing, where you identify the exact point in the genome where you want to amend the genetic problems, but that still, from our point of view, at least for the epithelium, still has enormous problems of efficiency. We have to have a very high level of efficiency in gene therapy, but as far as the vectors are concerned, retro or lentivirus, you can't really control the point in which they insert. It's basically random inside the genome. This can generate problems of genotoxicity. But we've seen that genotoxicity is not linked to the type of vector, but to the promoter. In fact, in any case, it is tissue-specific, cell-specific, and in fact, even disease-specific. For example, with the hematopoietic stem cells using the same vector, you have genotoxicity in X chromosome linked immune deficiency, but not in the one linked to tyrosine deaminase deficiency. Our cells are particularly resistant to transformation, but so far we haven't noticed, in spite of the many safety tests that we've carried out, we haven't observed any tendency to any problems of that type linked to insertion. But we're all, that said, we're all trying to develop methodologies for the future to have more certain, more accurate, more targeted genetic work.
Buonasera.
Intanto complimenti per il suo lavoro. Poi volevo fare una domanda di questo tipo. Lei ha fatto vedere che...
And then I wanted to ask you a question. You showed that you use stem cells for the treatment of some genetic skin diseases, like that child, for example. Is it possible to use it also for some cancers, for example, like skin cancers, or where you can't use stem cells? Well, this is an important question again. So far, with gene therapy, we have been able to attack the monogene disease. That is to say, the disease which is due to one gene. Well, why? It's pretty obvious. Say that you have a disease that involves several genes. It's very difficult to have a gene therapy. So they're monogenic diseases. And so, even in pathologies where you know that there is a multifactorial cause, it's very difficult. As far as cancer is concerned, there are. In the past few years, interestingly, there have been approaches which are linked also to the advanced treatment and to cell treatment that exploit or use the immune system with the modification of some immune stem cells. The ones that are being studied now are the CAR T, that are the ones where we try to modify some stem cell, through some stem cells, the immune system, so that it will attack the cancer, which is a very different approach from the one we have, although it involves also gene therapy, but it's more cell therapy here. The answer is yes, but it's still underway. There are clinical trials, as far as I'm concerned, as for CAR T. Fiorella, correct me if I'm mistaken, but basically it's only on blood cancers, aren't they? Yes. But there's certainly research in this field. Thank you very much. No, one last question. Last question.
Per il suo lavoro, per la persona che lei. Vorrei chiederle, ho sentito
I have heard that the study of stem cells has also been extended to the Langerhans cells in pancreas for diabetic patients. I would like to know where it stands. There are several groups in the various worlds that are working on this in a very active manner. One of the most serious one is Doug Belton in the US and Harvard. He is trying to generate, well, he's already done it. He's been generating beta cells of the pancreas, starting from stem cells, which are pluripotent ones from the embryonic or iPS. He is trying to transfer it to the clinical level. A few weeks ago, I heard people talking about it because there's a problem of delivery, of how you can transfer these pancreatic islands that are attacked by the immune system. But the generation or the production of these cells by pluripotent cells has already been completed. In fact, there are groups who, through genetic modifications, have managed to do something which is in direct planning. That is to say, without going through the pluripotency phase, they are able to generate beta cells from alpha cells, again through genetic modification. As to the best of my knowledge, that it has not yet reached clinical trial levels. It's fundamental research, which subsequently will have to be tested.
Bene, grazie. Grazie a Michele De Luca.
Thank you very much. Thank you, Michele De Luca.
Arrivederci a tutti.
Grazie, arrivederci a tutti. Grazie, professor De Luca.